I see a bag of something.
All right, so we're going to talk about the human life cycle. We'll do a lot more life cycles in next semester, but this is a great place to start because you can all you can all relate to humans, I hope, at least somewhat, or to yourself. All right, so to start our human life cycle, I'm going to draw.
A castle.
No, that's a temple.
A balloon? A balloon with two different springs. Are they married?
Good work.
We'll get there. We'll get there. All right, so these are sexually mature adults.
So they're buried in the temple, right?
We'll get there, okay? Spoiler alert. All right.
You see Brandon.
So this is a male and this is a female, okay? And what are the ploidy of their cells? Two, good. They are diploid adults, okay? Except for in certain places. In the male, he has testes. And in the female, she has ovaries. Oop, I spelled ovaries wrong. And what's occurring there is some of the cells are going through the process of meiosis and forming egg and sperm. And after they go through meiosis, what is the ploidy of the cell? All right, they are haploid. Okay, so in this life cycle, we have part of their life being in diploid and the other half in haploid. Now, if a sexually mature male and female fall in love and get married in the temple, they may, when the time is right, go through a process by which the sperm and egg are coming together to fertilize, go through the process of fertilization. So through the union of sperm and egg, you have a half set of chromosomes and another half set combining to make a zygote, right, which is a diploid fertilized egg. And then through many rounds of mitosis and development and specialization and reorganization, they will eventually become sexually mature, or that zygote will become a sexually mature adult. And then it also will produce gametes, barring any complications. Okay? So this is the human life cycle. And bless you. And so now we're going to talk about the process of meiosis. So how do cells go from diploid to haploid? And it's a lot like mitosis, but there are some very specific differences. All right, so meiosis has 2 rounds of cell division, and mitosis had 1. So we're basically going through the same phases, but doing them twice. So we're ignoring pro-metaphase. It does still occur, but we're just going to ignore it for the purpose of meiosis. Okay, so the first stage is going to be prophase, and this looks very similar to what it did in meiosis, where you have light and dark areas as the chromosomes are condensing. They've already gone through replication and interphase. And so that's prophase 1. But what starts to happen with these chromosomes, so through the process of fertilization, you had a copy of chromosomes from the father, and a copy donated by the mother. And what happens is the homologous chromosomes, so chromosome 1 from the mother and chromosome 1 from the father, start to come closer and closer together until they are connected, like right on top of each other, okay? And this process is called synapsis. And what synapsis allows the chromosome to do is another process where bits of each of the chromosome actually switch. So part of the one from the father and part of the one from the mother actually switch sides. And that is called crossing over. So what we're doing here is we're mixing up the genetic information. So now none of the genes are strictly the mother's genes and the father's genes. Or sorry, none of the chromosomes are strictly the mother's genes on it or the father's genes on it. But now they are mixed up. Some of them have a bit of each. Okay? So in metaphase, what happens in metaphase? The chromosomes align in the middle, right? But they don't align the same way they did in mitosis. Okay, so rather than aligning on top of each other, they are going to align side to side. Okay, and you notice I didn't draw all the mothers on one side and all the fathers on the other side, because the actual way in which they line up is random. Okay, so and this is called independent assortment. Okay, so this further allows for genetic shuffling to occur. So not only do some of the genes jump from one to the other, so I'll draw that on this one here. But they also don't assort, in any order. It's all just random. Okay? And so you also notice that the centrosome, the microtubules, don't attach to both sides. They only attach to one of the chromosomes and one side of the chromosome, so that they can pull them apart as tetrads, so they don't pull the chromatids apart. Okay, which was different than it was in mitosis, right? So in Anaphase, what happens is these tetrads actually stay attached. Oops. And the tetrads then get pulled to separate parts of the cell.
Is there any particular reason why they're the same color?
I just didn't want to redraw them.
Gotcha.
But in reality, they're all mixed up.
Okay.
All right, so in anaphase of mitosis, we doubled the number of chromosomes. Did we double the number of chromosomes in this phase?
Doesn't look like it.
No, we didn't. They stayed the same. So we didn't separate the chromatids, so the number of chromosomes stayed the same. So this is still a diploid chromosome. Or, sorry, a diploid cell. Okay, if this one cell was diploid, and then we just cut that cell. All right, but when telophase occurs, and eventually cytokinesis will occur, what will be the ploidy of these two new cells? Half. What are each of these two cells going to be? Right, so now this one is haploid and this one is haploid. So they only have one copy of each of the chromosomes. And then that is after cytokinesis, that completes meiosis 1. But there's another round of meiosis which occurs. At the end of meiosis 1, we have two cells. And that round of meiosis is meiosis 2. So meiosis 2 is pretty much the same as mitosis. Okay, so in prophase, you have the chromosomes condensing. Right? In metaphase, they line up in the middle. But they don't line up like this. They don't line up side by side like they did in meiosis 1. Instead, they line up on top of each other. Okay, and if you wanted to add a little detail, each of these chromosomes is, a bit of one and a bit of the other. All right. And if you remember from last time, so this is haploid, this is haploid, but then in anaphase, what do we do to the chromosomes?
Apart and becomes diploid.
Exactly. We split the chromatids this time. So at this point, we're doubling the chromosomes. So we took one chromosome that had two chromatids, and we split it into two chromosomes that have one chromatid each. So then in telophase and cytokinesis, you get haploid again because you're splitting each of these cells in half. And that's how you get haploid gametes, have half of the number of chromosomes as an adult cell. And so at the end of mitosis, how many cells did we get from when we started with 1? So at the end of mitosis, how many cells do we get? 2 from 1. At the end of meiosis, how many cells should we have? 4. All right, I didn't draw it here. Each of those 2 cells are going to go through meiosis 2, so then we'll have 4 cells. But those 4 cells will be haploid, and they will be mixed up. So no haploid cell at the end of meiosis is going to be the same as the other one because of crossing over and independent assortment. Question?
No.
Yes.
So meiosis 2 is just repeating the process of meiosis 1?
Yes, except the chromosomes align like they did in mitosis rather than like they did in meiosis 1. You're forming the question?
Yeah, so then after the end of meiosis 2, does that just keep happening over and over again?
Nope. So then these develop into sperm or egg, depending on if it's in the ovaries or the testes.
I thought we were going over the process of baby carrying. Okay.
No. Yeah. We aren't going to go how babies are made. You can come to my office hours if you want.
But a mom and a dad love each other very much.
Okay, so what are some similarities between the two? Okay, they have chromosomes.
Similar prophase.
Okay, prophase is similar. Let's just say they have all the same stages. Okay, what else?
Your cells.
Okay, it happened in cells. Fun stuff. It's pretty general.
Is that
How-
the same stages?
Same stages, yeah, they both have PP mat. How about you have cell division occurring in both?
Yeah, good one.
Thanks, Blake.
Okay. All right, in meiosis 2 and mitosis, how did the chromosomes separate? Right, chromatids split. All right, both require replication to occur. Okay, if you don't have the replication of the chromosomes, it's not going to work. All right, that's a good list. All right, well, what about differences? It's easier to point out the differences. Yes?
Synthesis and meiosis. That's the difference.
Synthesis? Well, they both have to have synthesis.
Okay, then what's the thing where it transposes?
Synapsis.
That's right.
Good. Yeah, there you go. Yes. All right, so meiosis has synapsis. What else did it have? Because it synapped, what else did it do? Crossing over. And then what occurred during mitosis? Independent assortment. Mitosis doesn't have any of that, okay? Say not. All right, what else?
2 rounds of meiosis.
Good. 2 rounds of division. All right, so because of that, how many cells are at the end?
4.
Okay.
Versus 2.
4.5.
Okay, and then, right, so this results in haploid cells. And this results in Okay, good. All right, what else?
And mitosis, not the cells just opposite each other, and meiosis cell.
In the end, you have 2 identical cells. In the end of meiosis, you have
Unique cells.
4 unique cells. Good.
Metaphase, there's 2 rows of chromosomes in meiosis.
Good, yeah. So you could say,
Tetra.
all right, the chromosomes separate as tetrads in meiosis 1. And they align as, and then in mitosis, the homologous chromosomes don't align, they actually go on top of each other. So the individual chromosomes are all by themselves in the middle. Okay, anything else? Okay, that's a good list. That's you can start to tease apart more differences than that, but those are the basics. Good. All right, any other questions?